So I think there is still quite a lot to be done in terms of resolving the species limits.
Sorry, that's my little bird flying around.
About a year and a half ago, we did a video on how the genus of SANSEVIERIA, commonly known as snake plants, had been incorporated into the genus of Dracaena after genetic analysis. One of the researchers we spoke to, Isabel Laridon, had been doing work on the genus largely to resolve evolutionary relationships among the SANSEVIERIA Clade, to find out when they started to diversify, determine what were the ancestral characteristics of the group, and trace their biogeographical history. She and her colleagues had finally put the finishing touches on the peer-reviewed paper, so we decided to sync back up with her to see what the results were.
So I guess let's get started, because it's been a while since I've chatted with you, and we had discussions on, more or less the SANSEVIERIA being subsumed into Dracaena. This is not what your paper is particularly about, but just to read the title of it, Plastid Phylogenomics of the SANSEVIERIA Clade of Dracaena Resolves a Recent Radiation. So why don't you just tell me a bit more of what the goal of this particular paper was?
Sure. So we started this study at Ghent University as part of a master thesis of Iris. And one of the ideas was to help us understand better what the species diversity is like within the SANSEVIERIA Clade of Dracaena, among others, to help us identify collections which are in botanic gardens, because it's very hard to do so based on morphology alone. So the aims of the study were to get a better understanding of the diversity and evolution of the group, and also its biogeographical history and morphological evolution in that group.
And you had mentioned that, sometimes it's when you're looking just at the morphology alone of this particular clade. How are some of the ways that people had classified, this clade of SANSEVIERIA in the past based on morphology?
So previous authors, mainly someone called Jankowski, he divided SANSEVIERIA into 3 sections based on inflorescence morphology. And then he subdivided those sections into 16 informal groups using mostly floral characters and some leaf characters. But again, many of these SANSEVIERIA species are only known from living collections in botanic gardens. They don't always are in flower. So it's very difficult to identify them based even on those studies, on those morphological studies. So DNA data is really helpful to get a better understanding of the species diversity in the group.
And what did you find? Did you find that they tucked in neatly and fit in nicely to the way that he previously recognized the different groups? So I think it's he's had Cephalantha, Dracomina, and SANSEVIERIA. That's how the groups that he used in order to separate them based on inflorescences. Did you find that they fit neatly in those groups, or when you actually did your analysis that they were kind of across the board and all over the place?
No, so some previous molecular studies based on more limited genetic information had already indicated that likely those groups did not match the true evolution of the group. And our results confirmed that for 2 of the sections, so section Cephalantha and section SANSEVIERIA, those inflorescence types do not seem to match the evolution. So Cephalantha was shown in our results to have originated multiple times as an inflorescence type from the SANSEVIERIA type inflorescences. While Dracomina, on the other hand, that was shown to have only originated once. So section Dracomina is a good group. So those species with that inflorescence type are all sister to each other. They're all closely related.
And do you get the sense that the SANSEVIERIA grouping is how would you describe it, the base grouping, or how it maybe originally originated?
So it seems from our reconstructions of the morphology of the inflorescence that a plant with something that looked like a SANSEVIERIA type inflorescence is ancestral to the other 2 inflorescence types.
Got it. I think the last time we spoke, you were mentioning that you were doing a whole chloroplast genome skimming. Were you able to do that? Because I think I had mentioned, or I think you had mentioned, or when I was reading that you might have had some challenges with some specimens doing a genome skimming.
Yeah, so we did genome skimming, and that basically captures those elements in a plant cell that occur there in multiple copies, which can either be chloroplast DNA because there are multiple chloroplasts in each plant cell, or it can be nucleoribosomal DNA, which also occurs in multiple copies. So we managed to reconstruct a chloroplast genome data set and also a nuclear ribosomal data set based on that genome skimming data. In terms of recovering the whole chloroplast, which is like chloroplast genes plus the intergenetic spacers between those genes to make a full circle of the chloroplast DNA, we managed to reconstruct a full chloroplast genome for one species. And then for the other 50 or so species in the data set, we managed to get most of that full circle. So our data set is pretty complete, but if we would want full circles for each of the samples in the data set, we would have to ensure that there's a higher recovery of the sequence data.
And what actually influences that higher recovery? Was it the materials that you had to start with, or is it something that happens in the process when you're doing the analysis?
It's a little bit of both. It's your DNA quality and quantity at the start, and also the number of samples that are put on the sequencing machine. Higher costs is higher data recovery. So we have to make a kind of balance somewhere. Our study was really aimed at being able to build a phylogeny based on sequence data. So for us, having full chloroplasts was a little bit less important than having like maybe 99% of the data for most of them. So we could actually run the phylogenetic analysis.
And maybe I should have mentioned this in the beginning, but how many species did you try to tackle, and did you try to tackle them across the various different groups, and how so?
So the SANSEVIERIA Clade is about 80 species in total, and we managed to include nearly 50 of them in our data set. But we sampled the data sets in such a way that the 3 sections and the 16 informal groups that were previously described were all represented within that sampling.
Oh, interesting. Okay.
Because we wanted to test whether that classification based on morphological data matches with what we get from the molecular results.
I know there's been some research in the past about how the SANSEVIERIA Clade has radiated outwards, maybe even how old it is. What did you see from your data analysis?
After we reconstructed the phylogeny, we did a dating analysis, and it showed that the SANSEVIERIA Clade is about 3.4 million years old, which is actually younger than most of the other succulent plant groups, such as cacti, for example, or agave. But it does match with a time between the Pliocene-Pleistocene transition, which included an aridification of East Africa. And our biogeographical reconstruction showed that SANSEVIERIA Clade likely originated in East Africa or Southern Africa. So that could kind of match, because the SANSEVIERIA Clade, one of its differences from Dracaena in a strict sense is that they have succulent leaves. So that could have played a role in their evolution.
And remind me, are there any SANSEVIERIA that are kind of more towards like the Arabian and also Indian area? And how do you think that those radiated from there? Is it just like birds traveling, or what could there possibly be there?
So the fruits are berries. So bird dispersal is definitely an option there. And we do see that there are species that occur in East Africa, for example, that have migrated to the Arabian Peninsula. So there are 2 dispersals to the Arabian Peninsula from Africa. There is just 1 dispersal from Africa to the Indian subcontinent, about 1 million years old is the estimation for that. So very recently. And in our data set, there is only 1 dispersal to Madagascar, but as I mentioned, it's not quite complete. We only have about 50 of the 80 species. So having all of the species included could potentially resolve another transition there. But that is what our data shows at this time.
When you were actually looking at this data, was there anything that you discovered where you're like, oh, this is actually the same species, or this is really closely related to this, or I can't resolve this through this phylogenetic tree because these look like a possible hybrid? Like, were there any of those bits of information that you could reveal to us from your data set?
So our data set is mostly geared at answering questions a little bit above the species level, because we wanted to check whether that classification matched what we're seeing from the molecular data. However, most of the species in the Sansevieria clade, the extant diversities of species that are there now, are less than 2 million years old. And in plant age, that's very young in terms of species. So many of the extant species are very closely related. Many of them are also only known from one location in the wild. So I think there is still quite a lot to be done in terms of resolving the species limits.
Sorry, that's my little bird flying around.
So that there's still quite a lot of questions that we need to answer. Our study was mainly performed based on samples from botanic gardens, public and private collections, and we really need to add more samples that are wide collected, preferably from the type locations, to really resolve those species limits. But we already showed that we included a few species with multiple samples per species, and there are clearly issues there with species limits. So we need to look at that in a lot more detail with more sampling.
Now, you said you concentrated a lot on the botanic gardens and private collections. Do we know if any of these plants are actually even in their locations any longer, and the type specimens and their specific locations? Are there any kind of data set or information about like conservation status of Sansevieria Clade?
That's actually a really good question. Currently, there is only one species which has been formally assessed on the IUCN Red List. There's also a species that's been assessed nationally in South Africa, for example. But most of the species have no formal assessment of conservation status, and this would be absolutely crucial in order to be able to protect them and include them in policy for conservation. But there are indications that some of the species have already been lost at the type location in Kenya, for example, and also that there are threats to several other species that are collected, that are in trade.
So do you think that you and your students are going to continue on this trajectory in this particular group, or do you think you're going to move on to something, or have you moved on already to another project?
Well, my core research is on Cyperaceae, on sedges. But yes, if I can find funding, which is quite important, this would make an interesting group to continue. I'm part of the Africa team in Royal Botanic Gardens Kew, and so being that Sansevieria Clade is mostly distributed in Africa, it would make a nice research project to continue. But then we would need to be able to do a lot more field work, I think, to collect samples from the wild instead of using living collections.
Amazing. Well, thank you so much for sharing some of this information. I know it was something that I wanted to include in the last video, but you just weren't quite there yet with the research. But I really appreciate this extra effort that you did, and some of the information that has been revealed to us through this study.
Discussing some of the latest scientific research out on peer-reviewed botanical and horticultural journals is not new to Plant One On Me, but we don't tend to do a lot of it. If this is a format you enjoy, you can let us know in the comments below. Otherwise, we hope that you'll consider subscribing to the channel to help support us produce more great botanical content. And if you're looking for information on outdoor gardening and landscapes, home renovation, and more, you could check out what we're doing on our new YouTube channel at Flock Finger Lakes. See you in the next video.